I'm Dr. Dan Simionescu, and I know bioengineering. Each and every disease we're trying to make human tissues in the laboratory. To do that, of course, we cannot use human components too much. However, we do combine cells, stem cells, adult stem cells, not embryonic, adult stem cells that we can collect from fat tissue, for example, which is available and widespread throughout the population. And once we take that fat, we can isolate adult stem cells, which are capable of transforming into almost any type of cell that we need for our applications. Once we have these cells, we combine them with scaffolds. Scaffolds are structures that, of course, the name implies a sort of a support structure, right? If you paint your house from the outside, you build a scaffold so that you could reach all points throughout the house. Similarly, tissue scaffolds are the skeleton of that tissue which cells hang on to. Scaffolds we develop by simply taking tissues from the pig and removing pretty much everything that is pig related, so that once these get implanted will not be rejected by the patient. And this process is called decellularization. It's another one of those words that scientists come up with that don't exist in the dictionary yet, but they will. It simply means taking out all the pig cells, so to prevent rejection, and removing minor components that can also induce rejection if it's implanted in a person. The next step will be to combine scaffolds with cells in such shapes that resemble the tissue that needs to be replaced. For example, we'll produce tubes that, once combined with human cells, they look like arteries. Not only do they look like arteries, they also function like arteries. They have the same elasticity. They have to be a little bit elastic to function well. They don't burst when you apply normal pressures and so on. So we do a lot of engineering tests in the lab on these products before we take them further.
So these vessels that you're looking at here, they're actually animal blood vessels that we have harvested, treated, and are now ready to implement into this bioreactor here. They would be mounted in the exact same fashion, and at this point, these scaffolds would be ready to have cells grown on them. So we would have them mounted, the cells would go through, the cells would grow into the tissue, and instead of water, we would have cell culture media, the food for the cells, flowing through these grafts in order to make them into living vessels.
Once we have established the perfect solution here in the lab and in collaboration with clinicians, we will prepare the products, the arteries, the heart tissue, whatever that will be, for future implantation into patients. And this way, the cycle gets back to the main focus of our work, which is the patient.